Okay, good afternoon, everyone. We will continue with our lecture on translation. And in the last class, we have learned about initiation. And in that, we learned about initiation factors in prokaryotes and eukaryotes. Eukaryotes one is complex. Prokaryote one is actually little simpler. All you needed was IF1, IF2, and IF3 in prokaryotic translation, whereas in eukaryotic translation it is complex. You need many initiation factors, and they would have been called as EIF. E is for eukaryote, IF is for initiation factors. If it is prokaryotic initiation factors, we would just call them as IF. And we learned about polyribosome, how the initiation happens, and in eukaryotes we learned about the importance of 5' cap for recognition recognition of mRNA by the ribosome, and so on. And we also learned about the circularization of mRNA, on which the ribosomes would load on one place from the ribosome binding site. This is kind of, you can say it as the ribosome, the processing of mRNA for or preparation of mRNA for translational initiation. Then you have the binding of the small subunit, that is the 43S pre-initiation complex, and then the binding of the large subunit, and then formation of the initiation complex. And then you can have the ribosome keeps moving, and while it is moving, it is reading the codons that are present on the open reading frame. Based on that, tRNAs come and keep moving out, while the protein chain, the polypeptide, keeps growing, or polymerization of amino acids keep happening based on the sequences of the codons that are present on the mRNA. And eventually there is stop codon, where the ribosome will dissociate, mRNA will retain like this, and the new, because of this circularization happens between the proteins, the initiation factors that are bound in the 5' side of the mRNA with that of the poly A tail present on the 3' of the mRNA. So there is an interaction between EIFG and these poly A binding proteins, and that keeps it circularized. So what we are going to learn today is, say we assume that we learned until start codon or the initiation. You wouldn't, if this is the start codon, you wouldn't have as many polypeptides, right? Say, for example, you did have had only one amino acid or two at the most. Anyway, we are going to learn about elongation, and hopefully we will finish until termination. If you have read the textbook or visited the textbook, you will notice that there are so many details and additional points that go beyond our syllabus. And not that we shouldn't go, but I think it's important for us to finish the syllabus first and then get back to those points if required. Even then, if you ever are interested, you can always approach and we can have a discussion on that anytime. But I'll try to touch upon most of this stuff. So as I indicated in the first lecture when I was starting on translation, I told you that the regulation part of the translation we will discuss along with in the next unit, where we'll focus on regulation. That will anyway come in the next chapter, so we'll not discuss much of regulation in this lesson. So we learned about some variations in the initiation, that is UORFs. I requested you all to visit the textbook. I hope you have done, and even if not, we will see how it is involved in regulation. UORF stands for upstream open reading frames. This is the primary open reading frame that actually produces a functional or structural protein. Upstream of them, there are very small open reading frames. Those are called as UORFs. So they somehow kind of initiate the uh, translation, and then the small subunit doesn't get completely dissociated. Instead, it keeps on moving until, and the initiator tRNA might bind to the P site, and then it will keep on moving until the start codon is encountered. And once the start codon is encountered, you will have the formation of the initiation complex, and translation of the primary or open reading frame happens. And I asked you to find out what is the significance of it. It comes as a regulation, and we will study in the next chapter. We also learned about internal ribosome entry site. One of it is about it. The 5' side of the mRNA folds into certain structures. These are all these alternate ones: upstream open reading frame or IRES. They're rare things. IRES you will find mostly associated with viruses. And if we study the molecular biology of viruses, there are so many diverse types of viruses. I think if we study viruses, we will find so many astonishing genetic elements and ingenious ones. Like in replication, we learned about priming protein. I hope you remember. DNA polymerase cannot initiate replication without a 3' OH. So there is one of the viruses has priming proteins. Similarly, you have these IRES, internal ribosome entry sites. Here, the IRS folds in such a way that typically the mRNA would require a 5' G cap, and E is bound to it, and then you have EIF factors, including EAFG. In this case, the IRES folds in such a way that it already can interact with EIF4G, and 4G binds here, and that is important for the binding of the 43S pre-initiation complex to the 5' side, and then it will scan for the start codon, and it will initiate translation. I mean, the large subunit will form, and then initiation of translation would have happened. So it is bypassing, this IRES is allowing the bypassing of the requirement for 5' G cap. And that is how it is helping. The other one is also interesting. There is another example of internal ribosome entry site, where the 5' end of the mRNA here folds into a structure that is similar to that of, or mimics the structure of tRNA, based on which the ribosome binds and forms something like this, and then translational initiation happens. That's something also amazing. So in today's class, our focus is on elongation, and also hopefully we'll get to termination as well. In elongation, our focus will be on several aspects. One of it is the mechanism of elongation itself. In that, we are supposed to learn about peptidyl transferase reaction, translocation of the ribosome, and more importantly, binding of charged tRNA in the A site. So we were in this place. The initiator tRNA is in the P site, ribosome, small and large subunit are set, and that is where initiation has happened. Now we need the other amino acids to come. And to come, they should come along with in the form of charged tRNA. And charged tRNA, so one is binding of charged tRNA. And we also have to ensure that the interaction between the codon and the antisense, or the anticodon. So what is present on the mRNA is codon. What is present on this one is the anticodon. The interaction between codon and anticodon should be correct. So we should ensure that the proofreading capacity of the mRNA. So Binding of the incoming charged tRNA is first. Ensuring that the codon-anticodon interactions are correct is the second one. Third is the reaction between these amino acids, that is the peptidyl transferase reaction. And the next part is the movement of ribosome by steps of in step of threes. Right? Those are the some those are it's a elongation is has too many activities, and it's a wonderful way of how ribosome does that. We are at the process of how the sequence, the nucleotide sequence, is now changing to amino acid sequence. So that's what we will learn in this. Translocation is the name given to when the ribosome, in the previous time we had here, for example, you have in the P site, the initiator tRNA is there. Now the tRNA is in A site. That means the ribosome has moved by 3 bases. That process is called as translocation. We also use the same word, translocation, in somewhere else. If you can recollect, we use that in telomerase. Right after synthesizing a little bit, the telomerase moves a little further to synthesize more. There also we use the word translocation, as it is literally it means movement forward. Right? Okay. Try to also remember when we are reading about elongation, we have to try to use this terminology: A site, P site, and E site. Amino acylated tRNA, that is where the tRNA is entering into here, right? Amino acylated tRNA or charged tRNA is coming in there, so we say it as A site. Peptidyl tRNA, often you will find the peptidyl tRNA in this place, like in here. So that is where the peptide is growing, right? So you call it as P site. And then E site, this tRNA will move away, exiting site. Okay. So there is how the first thing we will learn is how the tRNA is coming into the A site. And first of all, what should come? What should come is you have just tRNAs and you have tRNA charged with an amino acid. I'll just put it as AA. Okay. We don't want normal, uncharged tRNAs to come into the A site. They are not useful and they might be obstructing the process of translation too. So only the charged tRNA should come. Second important thing, what we will learn in the next slide, is we want the correct charged tRNA to come. Right? If there is a codon, we don't want any random charged tRNA to come into the A site. We only want the specific correct one to come. So whether it is correct or not we will deal with it in the proofreading mechanisms. Right now, the question is between these two. You have so many uncharged tRNAs, and you also have charged tRNAs. We only want the charged tRNAs to come into the A site. This site can actually accommodate any of the tRNAs, charged or uncharged. How can it happen? That is based on another factor called as EFTU. E is for elongation factor. EF is for elongation factor. It's called TU, EFTU. It is also a GTPase. That means it has a GTP binding domain. It has a GTP binding domain, or you should say it as GTP binding protein. So this EFTU binds to the charged or amino acylated tRNA. It does not bind to uncharged tRNAs. And this EFTU, when it is bound to GTP, that's in its active state, will bind to charged tRNA and then will bring that tRNA to the A site. Okay. So if GDP is bound, then the EFTU will not have affinity for tRNAs. So what happens as a mechanism, you can think of it, EFTU here along with GTP is bind to a charged tRNA, and it is now occupying the A site. Once it is bound there, you see if here there is one site that is called as factor binding center. EFTU fits into that center here like this. And when everything is fine, interactions between codon, anticodon, and many other things, then the factor binding site will stimulate the GTPase activity of EFTU. And GTP is hydrolyzed to GDP and PI, inorganic phosphate. And now EFTU does not have any affinity to the tRNA. So it dissociates and will move out. Okay, so that is about EFTU. Now what we are saying is now we have a problem is we have EFTU and it is now bound to GDP. In GDP state, it is not functional. So we need to replace this with, you should remove it and add a GTP. For that, we will need additional additional factors, that is EFTS. Maybe it will come in the other slide. I might have misplaced, but I will get to that. So here are proofreading mechanisms. When does this GTPase activity when is the GTPase activity activated, right? As I was telling you, the interactions between codon, anticodon should be appropriate. A typical ribosome, especially this is all what we are discussing about elongation, is when in prokaryotes. Eukaryotes also have almost similar, a little bit variation can be expected. But so there are three different proofreading mechanisms how ribosome is able to to ensure that copying of the mRNA, translating the mRNA into proteins. Right, those three mechanisms. One of my favorite is this one, which is something called as, we will discuss about it gradually. So one of the mechanisms uses directly about the interactions between codon and anticodon. The other proofreading mechanism is if the base pairing is not appropriate, if the base pairing is not appropriate, EFT here in this sense, incorrect pairing of codon anticodon, then the EFTU does not fit into this factor binding center. If that is not binding here, then the GTPase activity of EFTU will not be activated. Then EFTU will keep on binding to this tRNA. So no hydrolysis will happen, and then it might be dislodged out. Whereas if the binding between codon and anticodon is correct, then the EFTU fits well into the factor binding site, and then hydrolysis GTPase activity is enhanced, and then hydrolysis of GTP would happen, and then translation would proceed further. The third one is called as accommodation. I think I should use the actual web slides, but this is kind of introduction to the proofreading mechanism. So just hang on while I repeat it in case you didn't understand. I hope you can see that here the amino acid is in this place, or tRNA is facing this way, and this tRNA is facing this way. This is the amino acid, and the other amino acid is here. If the base pairing between codon and anticodon are fine, and the EFTU is GTPase activity is done and eliminated out, there is a step called as accommodation, where you compare between these two states. Here the amino acid on this side, and now the tRNA has moved or turned it towards the other side, where the peptide and the amino acid are nearby here. That step is called as accommodation. If the base pairing is not appropriate, then the accommodation step is inhibited. If that happens, the tRNA will move out eventually. So that is how it's not about picking the right one. It's about eliminating all the wrong ones. If the interaction between codon and anticodon is not appropriate, those tRNAs will be eliminated. It's not, I hope you understand, right? So if you had an apples, and if you want to eliminate the, assume that you had the apples and some of them are bad, right? I'm just talking in terms of a strategy. When you are putting into the another one, you will allow anything that is good, but if it is bad, you are discarding, right? That is the strategy it is happening. It's not like I will only pick good ones and put here. That is one strategy. The other strategy is I'm serially taking every one, checking if it fits, it is retained. If it is not fit, it is eliminated. That's how evolution also works. Anything can grow, anything can survive. If it fits that conditions, it will survive or exist. If not, they will go extinct. So the first step, I was talking about proof reading mechanisms. We will see these again, little more or kind of repetition. And this is my favorite, so I would like to spend a little time on this. You can focus on this case. So this is 16S rRNA, and here you have the tRNA. This part, this loop is tRNA, and this is the mRNA. This is the GCA is the anticodon, and you have UGC, that is the codon. They are a perfect match, so no problems. And there are two adenine molecules within 16S rRNA. They are called, their numbering is A1492 and A1493. That is the serial the position of those bases in 16S rRNA. You can see here, typically the decoding center, you can call it, whether this interaction between the codon and anticodon is fine or not. So these two bases here, the adenine bases, if you see, they are facing inwards. You can also, when the interaction between codon and anticodon are fine, say for example, this is the mRNA and this is the anticodon loop. When their interaction is fine, the two adenines flip out like this, right? They were actually inside, now they are flipped out like this. This will happen only if the interaction between codon and anticodon is good, or it's the perfect base pairing like this one. If the base pairing is not good, then the two adenines, okay, like shown here, they do not flip out and interact accurately. And then that is where it prevents the elongation to happen any further. There is an antibiotic. I also asked you to have a look at antibiotics. So I will just discuss this antibiotic here. It's called as paramomycin. That antibiotic targets translation, and it targets these A1492 and A1493. It lodges itself, this red one that is shown here is the antibiotic. It lodges itself within the foldings of 16S rRNA like this, keeping these two adenines always out like this, as if it is the correct reading. So what will happen? Normally, if there is no proper base pairing, these adenines will remain like this. If the base pairing between codon and anticodon are there, they flip out like this, and that will allow other steps to happen. That means the interaction between codon and anticodon is complete. But if you forcefully push out these adenines outside like this, even if the interaction between codon and anticodon are not good, this decoding, these two adenines, because they are flipped like this, ribosome, the proceeding reactions will happen. As a result, ribosome makes many mistakes. It's called as misreading. Why is that misreading happening? Because of this antibiotic has occupied this place, resulting in flipping out the 2 adenines like this. Okay, I hope you understood. If not, we will discuss in some other class as well. The second one is the GTPase activity that we already discussed, where if the pairing is correct, these two pairing is fine, codon and anticodon, then the EFTU will fit well into the factor binding site. And then GTPase activity and EFTU is released, and then accommodation and other steps would happen. But if the pairing is incorrect, this one is not appropriate, then EFTU will not have proper orientation to fit exactly into the factor binding site. If it is not, then it cannot have the GTPase activity activated, and then the translation is kind of halted. In that case, sometimes it will also come out like that, and then it will give a new opportunity for a better or a good or the right charged tRNA to enter into the A site, that is eliminating the wrong bases, wrong tRNA. The last one is accommodation. I hope you can see this clearly. Here is the amino acid tRNA, and it is oriented this way, and the amino acid is present in such a way. Assume that EFTU, everything has happened and gone away. Now, accommodation is the tRNA will turn a little bit, conformational changes, because of which the amino acid is in close proximity with this peptidyl P site. And that is called as accommodation. But if the base pairing between codon and anticodon is not correct, then accommodation will be prevented. So based on these 3 mechanisms, ribosome tends to eliminate all the wrong incoming or mispaired codon-anticodon sequences. There are alterations in the sense Wobble hypothesis, and there are also, we will discuss that in a later class. And there is also one more thing you need to remember is, despite these mechanisms, mistakes do happen. No process in biology is perfect, right? There will always be errors. But these are mechanisms to minimize these errors. So once ribosome assures all these kinds of errors and tried its best in a way to get the correct tRNA or decoded the mRNA properly, then the next one is peptidyl transferase reaction. Accommodation, as I've shown you in this case, is essential for peptidyl transferase to happen. Otherwise, it will not happen. So we already saw this reaction before, where we were discussing about all the introduction and components chemistry required to understand translation. So I'll just say it again. So ribosome, tRNA, it's in the peptide P site, and this is in the A site. Accommodation has happened, so tRNA has turned, and now the amino group here of the incoming amino acid will form a bond with the carbon, and one hydrogen will be given away till here. So OH will come here, and this tRNA is now free. And the peptide is now linked to the tRNA that is present on the A site. But most of it is in the P site here, like this. Okay? CONH. That's peptide bond. If you are going for any interviews after BTech or even during vivas that will happen during practicals and so on, you should be very thorough with the first 5 or 6 chapters of biochemistry. The typical to ask questions about amino acids, proteins, pKi, isoelectric point, and so on. So try to be good with those. Any interview, try to read those first 6 chapters and the techniques part as well, like electrophoresis, chromatography, and so on. If you know that, people will assume that you know a lot and you are trainable. That's the first indication. If you didn't know those, then you are untrainable. So keep that in mind. Okay, so we discussed about class 1 and class 2 in tRNAs. In class 1 you have the interaction. This is the, you have a sequence CCA, right? Adenine is here, so I will remove this adenine. um, I think they are putting it as, this is CC, okay? CC, that is cytosine and adenine. You remember that in the, there is a CCA enzyme that adds during the processing of tRNA. The 3' end of the tRNA is added with the sequence CCA. And I'm talking about the ribose of the adenine, the terminus adenine. It has 3' OH and a 2' OH. Okay, and there, so what happens is the amino group, the 2' OH happens, sorry, the 2' OH, this is the 2' OH. There is a role for it, and it is supposedly, it will interact with the, this is the amino group, okay, of this amino acid here. And here we have the peptidyl chain that is growing, and its carboxylic group. The peptide chain is attached to the 3' OH using the carboxylic group. So the 2' OH of the adenine is thought to interact with the, this hydrogen of the amino group, and so the electron, it will, the nitrogen here will interact with the C or the carboxylic group. That is how you will form a bond CO and this NH. That is the peptide bond. And a hydrogen from the 2' OH is given to this one. So it will become OH, as is shown in this case. If you see, this hydrogen actually belonged to the 2' OH. This hydrogen that is now present on the 2' OH actually belonged to the amino group here. So catalytically, 2' OH has a role in orienting or making electron transfer between the amino group and the carboxylic group, allowing the formation of peptide bond. Okay. So the next step is translocation. As I already discussed, translocation is movement of the base, a movement of the ribosome by a codon. A codon is 3 bases. For translocation to occur, we have a factor that is called as EFG, and it is also a GTPase. It binds to a GTP molecule, uses for its activity, you need GTP. So EFG has 2 conformations. EFG, when it is bound to GTP, it has a conformation something like this. And when it is bound, when the GTP is hydrolyzed to GDP, then it has a different conformation. We'll get to that. So once the peptidyl transfer reaction has occurred here, the EFG will occupy the little bit of empty space that is present in A site, something like this one here, if you're able to see. And then it will enter there, and the factor binding site will activate the GTPase activity, and GTP is hydrolyzed to GDP. When the EFG bound to GDP will change its conformation, and it is like it pushes the tRNA towards the other side. Actually, what happens is the whole set, the mRNA, one second, I'll try to use the whole mRNA along with the tRNAs are pushed towards this side by a codon, and that is done by the energy provided by the EFG. And how is that energy? It's kind of mechanical energy. GTP is in the form of chemical energy. The hydrolysis of it creates the moment in the form of change in conformation in EFG, and that pushes these all mRNAs and the attached tRNAs by a codon like this. And that step is called as translocation. If you see, there is something they call it as ratchet, meaning that you have these two subunits, small subunit and large subunit. They tend to, I don't know if I have it here. I don't think it's not there. I will try to get it for the next slide. There should be a small GIF file where it's like it keeps moving. These large subunit and small subunit, they are bound, but they're not static structures. They keep moving a little bit one on the other side, like a click, little bit moment of opposite moments. That's called as ratchet moment, and that is done during translocation. Once it moves, all this whole subunit, the small subunit moves here, and everything, the those that are present in the large subunit, sorry, the mRNA and the tRNAs are moved, or you can say the ribosome moves forward like this. And eventually the already existing tRNA in the E site will move out, and a new tRNA will come into the A site. So again, the process of binding of EFTU will bring a charged amino acid. Proofreading will occur, and then the testing, proofreading. There are three mechanisms, and once they are all done, peptidyl transferase reaction occurs. When that is done, translocation is mediated by EF gene, elongation factor gene. So there is something I didn't mention. Maybe I will discuss in the next class, or along with if we ever get to discuss about coronavirus. There is called as frame shifting, rare frame shifting tRNAs. That means they shift. If you are typically, elongation should happen in steps of 3 bases, because that's one codon. If it happens less than 3 or greater than 3, then the reading frame shifts. As I said, in some of the viruses, also in coronavirus, you'll find frame shifting, and that we will discuss some other time. Okay. So we are having EFTU. That is the one that is the EFTU is the factor that is bringing the charged tRNAs to the A site, and then it is getting consumed, meaning that GTP is hydrolyzed and GDP is released. Now EFTU is bound with GDP. With GDP, it doesn't have any affinity for charged tRNAs. Then we need to replenish or bring back the active forms of EFTU. Active form means it should be bound with GTP. So we need to exchange the GDP with a GTP. So the factor that does it is called as EFTS. EFTS, the function of EFTF is to remove or dissociate the GDP from EFTU. If GDP is removed, then GTP will occupy that place. If GTP is occupying the EFTU, now it is in active state and it has high affinity for MNSL tRNA, it will bind to it, and that can then be carried to the A site of ribosome. And then translation keeps on happening. Say, for example, if you have invented a drug that will inhibit EFTS, activity of EFTS, what will happen? Translation will come to a halt. Why would that happen? Because if EFTS is not there, there is nothing that can replace the GDP with GTP. If there is no EFTU GTP, then there is no way of charged amino acids, MNSL tRNA, getting into the A site. If that is not there, translation will come to a halt. Right? There are several steps or structures within translation that antibiotics are made for. Several antibiotics that are naturally produced by various organisms, they target translation because it's an essential feature, essential thing, like similar to that of replication or transcription also. But translation is much more easier because it's in the cytoplasm and it is continuously happening. And if you stop translation, the ribosome, the whole organism goes into kind of dormancy or even death also. So there is a point called as a concept called as molecular mimicry. You read about biology for engineers, and that is the objective of that course is to instill some kind of motivation or orientation so that you will be able to view the concepts of biology and mimic those concepts into to solve real life problems. Nature has been doing it all the time in that way, in the sense of at the level of mimicry can happen at the level of molecules also. What we are seeing here is the color magenta, or say dark red one, is the tRNA. This is the CCA sequence, and here you have the anticodon loop. So if you see the same tRNA, this is the tRNA in the green ribbons, and this part of it is EFTU. Okay, elongation factor TU. The structure on this side that I'm encircling is the structure of EFG. I hope you will be you notice that they have very similar structures, right? That is basically what is mimicry. I mean, you know, the A site of the ribosome, it has a specific shape, and those that fit into the shape will only be able to enter in, otherwise they will not be able to make proper interactions. tRNA along with EFTU will fit into this shape, and the next step after peptidyl transferase reaction is translocation, and that is done by EFG, and it has to fit into the A site to make translocation happen. If it has to fit, then it has to have a certain structure, that is structure complementary to the space available in the A site. So many proteins that you will find also in RR, ribosome release factors and so on, they also have similar structures like this. That is called as molecular mimicry. So I will review quickly, and then we will go to the termination. Okay, this is the overall picture of how you can see translation elongation. A site, this is from a research paper. It shows elongation. First step is charged tRNA binding. Second is peptidyl transferase reaction. In charged tRNA binding, we also read about proofreading mechanisms. Upon that, we have peptidyl transferase reaction and then translocation. Okay, so EFTU will bring the tRNA to the A site. Here you have the A site is empty. EFTU has brought the tRNA. It is now here. And GTP will be hydrolyzed to GDP, because of which EFTU will be eliminated. And then the accommodation. What is accommodation? The tRNA, the amino acid is, tRNA is placed a little bit outside like this. Now it is oriented towards the inside, if you see it here. That step is called as accommodation. Then we have peptidyl transferase, that is the transfer of this peptide to the A site tRNA. And now GTP bound EFG, the factor responsible for translocation, will bind here. The GTP will be hydrolyzed to GDP, GTP hydrolysis. That energy will be used to push the whole mRNA along with the tRNAs by 3 bases in this way. As a result, you can either say the mRNA moved or the ribosome moved. And now once the translocation is over, the EFG is released out. And then this step keeps going on until a stop codon comes by, right? Those are the essential mechanisms of elongation. In proofreading, we learned about decoding. We learned about A1492 and 1493 bases, 2 adenines that check whether the codon anticodon are base paired properly or not. Second is fitting of the EFTU into the decoding center, factor binding center, which is responsible for GTPase activity initiation. That is one factor. The third one is accommodation that we have discussed. If the base pairing between codon and anticodon are not appropriate, then one or more of these mechanisms will eliminate the tRNA. But nevertheless, mistakes do occur. There is a very low degree of translational errors. So termination, I'll try to give an introduction to this and then I'll stop. We'll take this in the next class. Termination happens when the stop codon is present in the A site. There are no tRNA that recognizes stop codons. Stop codons are UAG, UGA, UAA. There are two classes of release factors, and in prokaryotes, so release factors class 1 and class 2, they're saying. One of it is the class 1 are the ones that recognize the stop codons, RF1, RF2, RF1 and 2, and ERF1. In eukaryotes, it's simple because they have only one ERF1. That means they recognize all the stop codons. In prokaryotes, there are two RF1 and RF2. So RF1, UAG is recognized by RF1, UGA by RF2, UAA by both RF1 and RF2. So if there is a stop codon that is present here, then it will, the RF1 will bind to this site. And it is thought that there is a small serine, proline, phenylalanine. That's this tip of the RF1 supposed to have something called as peptide anticodon, right? In the sense, say for example, this is UAG that is recognized by the stop by RF1. Okay, here I put UAG. That's the stop codon. The tip of the RF1 supposedly has serine, proline, phenylalanine. Those are thought to have some kind of interactions, or it is thought that they recognize these. This is called as peptide anticodon. It is, we don't know exactly. I think still there might be some variations or additional factors required, but it is thought that this release factor 1 is able to recognize the stop codon using this peptide anticodon. You call it as peptide anticodon because these are actually amino acids and they are recognizing a stop codon. So they call it as peptide anticodon. And there is also, termination has three important factors, three important events to do. One is to recognize the stop codon. Second thing is to release the peptide. This peptide should be released here. And the third thing would be that the ribosome should be released. Only then these ribosomes can initiate translation again somewhere else on some other mRNA. So for the moment, what you will remember is that there are two classes. Primarily, we'll discuss only RF1. RF1 and RF2, they are class 1. They are responsible for recognizing the stop codons. And at least RF1, it is thought to recognize by using a peptide anticodon. And RF3, the second class 2 release factors, they bind to the RF1, class 1 RF1 or RF2, and then they stimulate they exchange for GTP, and in GDP state they are not as active, but once this GDP is exchanged for GTP like this, then they become active, and then the GTP is, because of that change in conformation, because of binding of GTP, they will allow the dissociation of release factor, class 1 release factors. And eventually, the GTP is hydrolyzed to GDP, and RF3 is also eliminated. We will have ribosome recycling factor that we will discuss in the next class. But I hope you got some introduction to termination. You have to remember class 1 and class 2. Class 1 release factors for recognizing the stop codons, class 3 to get the things done. What is that to get things done? To remove the uh, release factors once. Okay. Please read this chapter. As I was telling, this is the peptide anticodon that I was telling you. Okay, I forgot about this slide. This is the stop codon that is here, and the peptide anticodon is thought to be here. These are the amino acids that interact with these bases. We will discuss more in the later class about GGQ and so on. Ribosome recycling factor, this is here. And yeah, please have a look at this antibiotics one once again. I need that you all have a thorough understanding of it. In the next, any of the biology or any techniques you use, you will, if you are working with living organisms, bacteria or anything, you will eventually use antibiotics, and you should know how they work. You should know the way to read and remember the antibiotics also.